A very good morning to all of you. Welcome back to the fifth and final day of the FDP and recent advances in biosimilar development. Today, in the first session, we have a speaker, Dr. Ashutosh Jani, who has done his master's in pharmacology and PhD from master's and PhD from LMCP, that is LM College of Pharmacy, Gujarat, and currently he is the head clinical operations department of Veeda Clinical Research, Ahmedabad, India, where he is overseeing to conduct of patient-based PK and clinical and endpoint studies, proof of concept studies, and late phase clinical studies for ANDA, 505(b)(2), EMA, and DCGA submissions, and oversees its execution. Earlier, he was senior general manager of clinical trials in Lambda Therapeutics Research, Ahmedabad, and before that, he was the head in the clinical trials department of Acute Test Research Laboratories, and also the manager in Lambda Therapeutic Research. He has an in-depth understanding of business process and structured methodologies and amalgamation of technical and commercial aspect of clinical trial business. With this brief introduction, I request Dr. Ashutosh Jani to deliver a talk on comparative clinical efficacy studies for biosimilars. Over to you, Dr. Ashutosh.
Thank you so much, Sundar, for the introduction, and it's a pleasure interacting with people around there. So I think you already had an enriching session. We are entering the last day of your FDP, and I'm sure you are have gained pretty much knowledge with respect to biosimilars, what they are, how they are made, etc. So what we'll be doing is we'll be touch basing basically onto the efficacy studies which are needed with respect to the biosimilars, and we'll be sharing some examples with respect to the same, and so to get on more clarity with respect to the design, how are they the protocols designed, or what are the things aspects with respect to the study which are required. So for all those attendees who are there, I think you could see my screen, and on the screen there is a pin. I would request all of you to get into the screen and just enter this game pin. Just go to www.kahoot.it through your mobile browser, or it could be through your whatever laptop or whatever device you are on. You could just log into, just go to www.kahoot.it and enter this pin. So I'm audible, right?
Yes, you are audible. We are.
Okay.
Yeah.
Thank you. Follow me. Thank you so much. I'm not sure if the previous speakers happen to use these type of platform or no, Sundar?
No. We have not used it. Yeah.
Okay. So that's why it's taking some time. Thank Kanika, Uma. Krishnendu. Come on, we have about 40 participants. We need at least 20 guys to start off, right? I hope it's not that early morning for you. It's already 10:15, so I think you guys should be in all of your best senses at this point of time. Thank you, Supreet. Thank you, Sid. So you're just 11. I need some more. Okay, so basic purpose of this is just to make it more interactive, because I know sessions over a webcam are pretty boring. Thank you, Prasanna. I don't know if anyone is facing difficulty. It is very simple. You just have to go to your browser, just type www.kahoot.it, and there'll be a screen which will be visible, and you're just supposed to enter this pin. That's it. Few more people. Okay, I think people are leaving. I'm not sure. Okay, I need some more people to start. Thank you, Divakar. Okay, Suraya, thank you. They say 13 is not a good number, so I need one more to start. I think probably you guys. Okay, one more to start. For those who just joined in, you just have to go to your browser, type www.kahoot.it, and enter this pin. Okay, I think people are leaving. Okay, thanks, Danny. Need two more to start. Of this 48 people, only 13 are able to log in. Thank you, Shilpi. Okay, so I think we are good to go now. Thank you, team. So what I would request is there'll be a question which would be displayed onto your screen, and you're just supposed to select whatever is applicable to you. So the basic purpose of this was I just wanted to know what is the audience comprising of. So whatever is the appropriate thing, you could just tick on the answer, and thank you. So we have 6 answers, 7, yep, 8, yep, okay, good. I think we have 30 more seconds if anyone has just joined in. Okay. So what's essential is it's important that we know each other, and I get a fair idea about what the audience comprising of, and based on that I could probably gauge my session, and probably we could run through certain things, we could stop at certain things, we could elaborate certain things, right? So good. So there are a good amount of teachers and faculties who are a part of the session. Thank you. Okay, Sanand Kumar was the fastest. I'm not sure if you know this, Herceptin. Have you heard of this? If you have heard of it, you could tell true, and if you've not heard of it, you could just tell false. Okay. This is just to get an fair idea about certain things which you know, you don't know, and that would definitely help in uh, gauging the lecture onto certain topics, right? Okay, so we have people adding on. That's good. Nice. Okay, so time's up. Okay, so good to know that a majority of the people are aware of what Septin is. So that's good. Okay, so with that brings me to the start of the session. So with this understanding, we can probably start off with the presentation, right? we'll go back to the quiz whenever you guys feel sleepy. Just to keep you guys awake, we'll again go back to some more questions. Okay, so thank you for being a good participant, and I hope you enjoyed that. So that brings me to my topic today, and what we plan to do. As I briefly mentioned, we'll be taking care of certain things wherever they pop up, right? So thank you for having me, and based on your answer, which the couple of answers which you replied, I just wanted to know, like, apart from the basic introduction which Chandra gave, so I basically am also a avid runner. So I do run marathons. Uh, uh, Marathons is my passion. So I run for soldiers and so on and so forth. The different marathons is what I've participated in. And I'm not sure if any of you been part of any marathons. So what they have happens is, like, at the end of the marathon, they do provide some snacks, which is complimentary. Obviously, that's free, and the snacks would be either some fruits, because of this unhealthy event. They provide some healthy fruits and some sandwiches or tea, coffee for post running of the marathon. So there was this one of the marathon, and myself and my other friend, we ran the marathon, and we were almost exhausted. And subsequent to that, we just went to this refreshment counter which was there, and we saw that there was a huge line. So there was a queue of people waiting to get their sandwich and fruit and tea, coffee. So I just told my friend that, come on, let's grab a fruit. So he said, no, this is a big queue. Why should we get into that line? I said, but it's free and we should have it. And he said, but no, it's a long queue and it doesn't make sense. I still insisted. He didn't turn up. So what I did is I just bent myself and tried to grab fruit, and what I got was an apple. So basically what happens is that there are two type of people. Some people are the ones who see what they want, and the other type of people are see something which stops them from getting what they want, right? So my friend was those type of person who see something which stops him from getting what he actually wanted. And myself, I made up a way and got to what I wanted. However, I was not able to get a choice. That I got an apple, I did not get a banana and orange. But yeah, that's what I had to settle out with. So as I mentioned, there are two type of people, and there are ones who like to get to whatever they want. And one of such person was this. Uh, he was a doctor by profession, and I'm not sure if anyone has watched this movie. The movie is Living Proof, and it's definitely based on a true story. So this is a story about one of the doctor who was the key person in getting Herceptin, the one which you just picked up on your quiz. So he came up with this, and this movie beautifully depicts his struggle and his sticking to what he wanted. As I mentioned, you have to get what you want. There are no people who could stop you from getting what you want. And this movie briefly depicts the same thing, that nothing stops you from getting what you want if you put up the best efforts. So on a personal front, I would recommend this movie. It's a story about Herceptin. For those who don't know Herceptin, it's trastuzumab. It's a biosimilar and monoclonal antibody which is used in breast cancer. So this was a special type of drug which was targeted for breast cancer, and it has done wonders. So the rest is history. So coming to some history about biosimilars, so they have been around quite some time, and the industry is gradually growing towards biosimilars, and the focus of the world is gradually getting on to from small molecules, which typically are they're moving on to large molecules. Those are biosimilars. I'm sure you guys will be having plenty of mangoes in this season, and all these mangoes are biosimilars, and so are cheese, bread, beer, to name a few. So we get Kesar mangoes, that's the famous in my part of the country, and the rest of the mangoes are biosimilar, almost similar in taste, and do the same thing. Something plus and minus, but still they are all mangoes, right? So these are all biosimilars. For me, my standard is say a Kesar mango. Probably you are from Maharashtra, your standard would be an Alfonso, and so on and so forth. But yeah, they are all ultimately mangoes. And so is the case with cheese. So we have Gouda cheese, cheddar cheese, all made up with slightly different microorganisms processed to form cheese, or a bread for that matter, or beer for that matter. So these are all different biosimilars which are there in the market, and we targeted from these food industry onto our drug industry, where we try to prepare various biosimilars by various methods and processes. Now, what brings to this biosimilar? Why are they hot? The main reason of them being hot is there is a patent expiry. Apart from that, there's a huge potential in a market. So nearly 17% of the drug spends are allocated to biosimilars. And there is definitely a growth in the sales across the world, especially when we consider biosimilars. However, that all these things come at a cost. So you need to spend a hell lot of money to get a biosimilar onto the market. It's not as simple as a small molecule. So that's the reason you don't find much players or many players in the country or outside the country who are focused on to biosimilars, because it needs a lot of investment, and everything is coming at a huge cost. So these are a few pipelines of biosimilars which are there, and that includes Herceptin, that is trastuzumab, what we were talking of, right? So these are various biosimilars which are getting expired during the course of the time, and the market would be flooded with their biosimilars in the near future. So what we was, I just tried to break down this presentation into various factors which are typically affecting the efficacy studies which have been conducted for biosimilars. So when we come to efficacy studies of biosimilars, so there are these key factors which are taken into consideration more from the patient perspective, the disease perspective, what you would like to see as an endpoint, and some design factors. So as I mentioned, there are patient factors, like say there are certain age which would like to restrict, or there are certain ethnicities which could be affecting the clinical efficacy studies. On the disease front, different biosimilars behave in a different mechanism of action. Their targets are different. You could also combine therapies. So there's a single therapy or a combination therapy of biosimilar cocktail sort of. And also certain endpoint factors would also be differentiating what you'd like to see as a short term, as a long term. So all these factors put together would be comprising for various factors for designing of the efficacy studies. And stratification is definitely one of the main criteria for the development of biosimilars. Now, when we come to this slide on background therapy, so what I mean to say is these are with adalimumab. I'm not sure if you know adalimumab. It's used for rheumatoid arthritis and psoriasis and so many other indications. So what happens is there are various companies who are preparing their biosimilars based on their pipeline of products. So they have been named differently, and if you compare the studies, if I'm say there's a study done by Amgen or Sandoz Novartis or Mylan or Oasis for that matter. So what are they doing is it's been quite challenging to recruit patients, especially when we come to say US or Western Europe. This may allow access to branded and biosimilar versions of an approved biological. So what happens is these are the countries in which there is already a biosimilar which is available. Okay, so if I design a study or if I am looking for some efficacy parameters and I conduct the study in say US or Western Europe, there are high chances that the patient would have received one or another biosimilar in the past, right? So when these studies, efficacy studies are designed, the same adalimumab biosimilar study being designed, so the Amgen would be allowing more than 2 agents, which is some biologics would have been given in the past. I'm going to say that patient, because you're conducting the study in a country which already has a lot of biosimilars in the market, there tend to be high chances that patient would have already received a biosimilar, which could not be the case when we go for certain developing countries or underdeveloped countries. So say Amgen's phase 3 biosimilar adalimumab trial that was in plague psoriasis, they will be enrolling patients who have received at least one or more than one therapy, because there are chances that the drug would be available and they will have to enroll those patients. So the likelihood of response to the subsequent treatment with the biological agents, they seem to decline. Now, say if I have already received one biologics or biosimilar, and then I'm not getting that response, and then I tried for an Amgen biosimilar, same adalimumab from another sponsor. In that case, there are high chances that the TNF inhibition, which we tend to get through adalimumab, we may not be able to get to that extent because the patient has already been exposed to adalimumab in the past. So if we compare, say, Amgen, Sandoz, Mylan, and Coherus, then you could see that the last one, Coherus, they did not allow any previous treatment. So if you want to go with this criteria, you need to go to a country where there is less or no biosimilar available. Only then you'll be able to get these things, right? On the same lines, when we come to masking, so what happens is it's difficult to mask the substances, and at times it's practically impossible, because say either one could be an injection, a pre-filled syringe. I think you all know what's a pre-filled syringe. It's already a syringe, and the other could be a vial. So now anyway, you could know that if it's a vial, it's this product, and if it's a pre-filled syringe, it's the other product. So masking, avoiding bias, these factors would really be challenging when we try to see the efficacy studies comparing the two biosimilars. So on the same lines, sharing with you the example of Humira. So that's the innovator, the first biosimilar adalimumab in the market. So that's the reference standard. Now, this Humira is marketed as a pre-filled syringe, and you could easily recognize that. And when you try to do a study with Humira, because you have to compare Humira with your generic or your biosimilar which you have prepared. So in that case, maintaining a double blind for the pharmacist would be difficult. So then we need to have a separate pharmacist who will be blinded, and there will be one which will be unblinded. Moreover, if it's a bi-weekly adalimumab injection which will be administered, then there'll be more visits which would be required. So there'll be, in case of a double blind, it would be easy to interpret whether the patient is on which arm. So these type of aspects, when you compare the efficacies, tend to be really important. The another factor which is important is stratification. Now, what has been reported is in terms of weight. If we see that, it appears that the efficacy is decreased mainly in patients with body weight more than or equal to 90 kg. Now, if you are overweight or probably more than 90, or with a body mass index of more than 30, that puts you in an obese category. Then a most significant decrease in efficacy occurs when the patients are in this body weight, or with which they have more than 140 kg of body weight. Thus, a same biosimilar adalimumab study, it might be worthwhile to consider while using the weight as well as BMI, so that to take care of the obesity, because it tends to decrease the efficacy. So if you have more obese patients lined up in your study, your high chances that your efficacy parameter would be affected. Therefore, the stratification is done. So what they do is, like, say, if you are enrolling one obese in a test arm, you will also enroll one obese in a reference arm. So you would like to stratify the people who are being part of the study. Similarly, there are certain disease activity scores. We call it as DAS. This is more used in case of rheumatoid arthritis. So since it's difficult to characterize the rheumatoid arthritis, how bad it is, we have this scale of disease activity index, and based on this disease activity score, we come to know that this is a severe case of rheumatoid arthritis or a mild case of rheumatoid arthritis. And based on these, the studies, means the patients from developing countries have often more active and severe rheumatoid arthritis, because there is no treatment. And as they may also show good responses, especially in case of placebo arm. So when we compare that to patients from, say, US or Eastern Europe, so when we come to developed countries, the response also changes. So if I conduct the study only in, say, India, or I conduct the study only in, say, Europe, or across Europe, US, and India, I need to ensure that there is a proper mix of patients which have been stratified. Say, if there are all the Indian patients are going into one study arm, they may skew the result, which will be the case. So this stratification is equally important. Also is important the geographical distribution. So distribution of the patients across geographies is equally important. Now you could see this. So this is with respect to the patient population, what we were taking into consideration. So there are different patient population which could be targeted for adalimumab. And adalimumab is also used in case of rheumatoid arthritis, psoriasis, psoriatic arthritis, ankylosing spondylitis, Crohn's disease, ulcerative colitis, plaque psoriasis. So these are various indications in which they tend to decrease the TNF alpha, and that's inhibiting of tissue necrotic factor. Now, the response to adalimumab therapy was lower in patients with, say, plaque psoriasis, who switched from one anti-TNF therapy to another. So the likelihood of response to subsequent treatment with the biological agent seems to decline with increasing the number of previous treatments with TNF inhibitors. So if the patient is already exposed to one TNF inhibitor and then there is again a disease progression, then the next TNF inhibitor may not be of much use, or the effect may be decreased. The another factor which also affects the design is the disease factor. So we'll be taking that into consideration. So this is just sharing with you the data, like various companies as we've seen. So there is Amgen, Boehringer Ingelheim, Fuji, Pfizer, Sandoz. So they are all developing their Humira. Humira is the innovator. So adalimumab is what they are all developing. So in the efficacy trials of the innovator Humira, at least 9 tender joints. So if you see somewhere here, we just listed out the disease activity. So you could see that there are more than 6 joints, swollen joints, different types of joints which have been involved, which should be present for a patient to be a part of the study. Now, what happens is when the innovator Humira was did a study, they involved at least 9 joints. So I would say you could understand the severity of the disease which they involved, and the patients were involved in. So at least 9 is what they considered as a tender joint, and at least 6 swollen joints were part of the patient enrollment. Now, when we see the other people who are trying to bring adalimumab in the market, what they are doing is they are enrolling only, say, patients with more than 6 joints. So 6 is fine. You may not go up to, the criteria, entry criteria is not as stringent as that of Humira or the innovator drug. So they're just involving 6 joints, so maybe 6 swollen and 6 tender joints against 9, which was the case with Humira. And apart from that, in addition, they are comparing to ESR. Now you could see that there are certain who are comparing to CRP, that is celiac protein or erythropoietin sedimentation rate. So these are biomarkers, so they give a fair idea about the level of the RA, the severity of the disease. Now, when you see that everyone has a different criteria with respect to CRP, and in addition, ESR, CRP is simple, validated, and a reproducible thing which would not be affecting based on your gender or age. So a CRP is a better marker as compared to ESR, because ESR depends also on age as well as the gender. So these things are to be taken into consideration when you are comparing the efficacy with different agents. So when we come to a biosimilar, there are a lot of battery of studies which needs to be conducted to get their idea about their pharmacokinetic, their biological functions, the structure, attributes, etc. Coming to this efficacy study of adalimumab, so the basic thing was to check the sensitivity of detecting the small difference. So if you are able to the disease indication, what we need to have. Say Amgen is doing a study in rheumatoid arthritis as well as plaque psoriasis, like PSO. However, Boehringer Ingelheim is only targeting rheumatoid arthritis. Now, if you target plaque psoriasis, there are high chances, say the responses are better when you use this indication as compared to rheumatoid arthritis, because a small change, even a slightest difference between the two products can be easily magnified when you are using the right target population, that is say plague psoriasis. However, if you go with this indication of plague psoriasis, you may end up conducting the study for years, because their patients, getting these type of patients is rare. I mean, so you get them, but they are not as many as compared to a rheumatoid arthritis. So then the company, certain other companies have selected their indication as say rheumatoid arthritis. So because that tends to have an advantage of larger patient population, so the study would be getting completed earlier, right? So with this also comes extrapolation of indication. So now what typically we do in small molecules is, if your drug, say for example, doxorubicin, you've done a study in breast cancer and it's been effective, the sponsor would tend to extend that indication from breast cancer to say ovarian cancer also, because it's also a type of a similar cancer. So these extrapolation of indications, you do study in one indication and then try to prove that this can also be effective in the other indication based on the label claims. These things are typically not possible, especially when we come to biosimilars. So these are all different regulatory authorities, say Europe, Health Canada, Australia, Japan, the US FDA. So what happens is, like say, the difference in the ability of the two products to induce this antibody dependent cell mediated differences, that is a really important thing. So based on the same only, we could be able to give an approval. So if based on the data, we've got an approval for rheumatoid arthritis, and you have not done any study in say Crohn's disease or other indication, the group, I mean to say the gastroenterologist who could be your potential prescriber, would not be comfortable with prescribing this drug in Crohn's disease, because they say there is no data for this particular drug in Crohn's disease. So when you come to biosimilars, extrapolation of the indications is not that easy, and certain regulatories do allow extrapolation, certain do not allow extrapolation. And even if there is an extrapolation of indication, there are certain groups like say European Cons Society, or say Indian Society of Gastroenterologists for that matter. So they may not be comfortable in prescribing adalimumab to their patients because of no data with respect to the efficacy. So in nutshell, you need to conduct studies for all the indications in order to get through that confidence, and obviously this adds on to the money. Apart from that, there are also cases with respect to interchangeability. Now, say for example, you already have taken a drug from company A, and the same drug, I'm going to say the same biosimilar, can you interchangeably used? I mean, say one day you're doing from company A, you don't get that drug available, but then the pharmacist at the medical shop would give you the same drug from company B, which we typically do for small molecules, right? For if you don't get drug from paracetamol of company A, you go for paracetamol of company B, and again after 1 week you get back to company A. So that's all interchangeability which you do, which is not the case possible when we come to biosimilars. And substitution, so if you are already on say drug A, biosimilar A, and you cannot be switched on or you cannot be substituted to the next biosimilar unless until your physician allows. So this switching is also important, like say patients from each treatment arm may be randomized to the reference product or a biosimilar, so that the part of the patient will be tailored. What happens is to ensure that the reference to the biosimilar product or vice versa. So it may be interchangeability is what we're targeting, that maybe you change them from one to another and the other to one. So you're just switching, interchanging them, switching them, which is typically not acceptable by certain regulatories. So there is a special study which was conducted, which was a North Switch trial. So where you tend to see that the disease worsens was defined as the disease specific components which were measured, and they were leading to major change in the treatment, like say rheumatoid arthritis, spondyloarthritis. These are all different indications which we already saw. So what is interchangeability? So we already have a reference drug and there is a biosimilar A available, there is a biosimilar B available, biosimilar C available. So you can switch from reference biologics to either biosimilar A or B or C. But when we talk of interchangeability, we are talking of whether you could switch from biosimilar A to B or B to C or A to C, whatever. So this interchangeability with reference is fine, but the interchangeability between each other, is that acceptable or is that a good idea? So this is what is essential, that you are aware that what happens. Okay. I think with this, you guys would be almost uh, falling sleepy, and we could then also switch ourselves from this sleepy mode back to our active mode. So if you are able to see the screen, we have one more question coming up. Okay, Nand Kumar is leading. So there's one more question. Have you heard about Hatch-Waxman Act? I think you guys are sleeping. We only have 3 answers. Okay, so gradually we see people waking up. That's a good sign. Okay, so there's a tie, yes and no. So not much aware, I presume. Let's go with a no. Okay, so Nand Kumar left. We have Prasanna topping up. Okay, so the next question is, if you are a physician, would you prescribe a biosimilar to a patient, even if the insurance company is not willing to pay for the original product? Okay, so I see people are waking up. That's a good sign. Okay, so for guys who joined us late, it's basically we're just on Kahoot.it, and you're just supposed to log into this. You could see the details here at the bottom. You just need to enter the relevant information. This is the game pin 6716665. For those who guys joined later, just log into www.kahoot.it, enter this pin, and you will see the same screen getting displayed onto your screen, and it's supposed to answer. Oh, it's a tie again. That's good. So Prasanna is still leading the show. Okay. Coming on to the next one. So for those guys who just joined in, you can log into kahoot.it. The game pin is 5716665. And you're able to log in. You will get this screen, and you could just pick up your answer, and we are good to go. Okay, good. We have more people waking up. I think it's tea time. Oh, good. Okay, good. Okay, 10 more seconds for those guys who just joined in. Kahoot.it, you'll get this screen. You just have to enter this pin 5716665. Okay, that's it. Good. So you trust small companies. That's good to know. So you would also be prescribing or giving drugs from small companies. That's nice. Okay, so that brings me to after waking you up, we're back to the presentation. So for those guys who don't know this, Hack Watchman Act is it's an act in US which allows that if you are an innovator, there should also be someone to compete with you, so that the ultimate aim is to get the drug at an economical rate to the general population. So that's what is essential, and for the same they have a law in place, and which is taking care that whatever the drugs are, I will say you could see that when there are prices with respect to small molecules after patent expiry, they drastically go down. However, with biologics, they do go down, but that's not that drastic. So the competition on the price is definitely there, but the drastic downfall which you see for the other small molecules, that's not possible with respect to biosimilars. One of the main reason being that every company cannot afford to produce a biosimilar. Okay, so this is adalimumab. So this is the innovator which is already there in the market, Humira. And then there were other biosimilars from Amgen, Boehringer Ingelheim. These are all big companies, but still they are producing biosimilars. So they are treated as biosimilars. So what happens is, like say, the two companies from Amgen and Boehringer Ingelheim, they would not get approval for, say, indication of, say, pediatric Crohn's disease, or for that matter, hidradenitis suppurativa, or non-infectious uveitis. So these are indications for which the regulator would not be giving them permission. So the innovator adalimumab, Humira gets in approval for all these because they have done studies in all these indications. Say bevacizumab. So bevacizumab is coming from Genentech, and then there is another bevacizumab from Amgen. Now, when you try to compare the various indications in which the bevacizumab is used, so given the indication, they do allow the use of the generic or the biosimilar from Amgen in all the indications except say epithelial, ovarian, fallopian tube cancers, or these are a special type of cancers for which you don't get an approval, right? On the same line, say Etanercept, we have Amgen is the innovator, but then Sandoz is the next company who brought Etanercept to the market, and they did all the studies and they get approval for all the indications which the innovator got. On infliximab, Janssen is the innovator, Pfizer and Merck. These are big companies. They have a biosimilar in place, but they don't get approval for pediatric ulcerative colitis because of the indication. This is in children. They have no data from the other companies with respect to the same. So these are comparative things when it comes to the factors which affect the efficacy. Endpoint is anyway one of the main one. So you should have a tough endpoint to get based on the regulator. So say for example, rheumatoid arthritis, you could evaluate the progress of the joint damage by an X-ray, but that's not tough. Okay, so you need something which is really hard endpoint, which would be helpful in getting you the exact differences. The sample size also plays a vital role in case of these type of studies, and the total study duration. Now, if you could see that this slide, we have the same adalimumab is what we are comparing. So they have these different efficacy endpoints, and the duration you could see is different for all the indications. So there is as low as 4 weeks. You do a study in Crohn's disease. You do it as large as say 52 weeks for psoriatic arthritis type of an indication. So there are various parameters which are important and which affect the efficacy of various adalimumabs when we try to compare them doing various studies. The usual design is a non-inferiority or equivalence is what. Equivalence designs are preferred from the easy point of view, and it requires acceptance by the regulators and can be directly affect the sample size. So the choice of equivalence margin is also an important parameter. So what endpoint would you like to take and how much should be the study duration? These all needs to be factored in. This is an example of Remsima or Inflecta. So they did the study with an equivalence margin of 15%. Okay, so what we need to see is at the end of 30 weeks, there should be a sizable difference of 15% for the ACR 20 score. So ACR 20 gives you idea about the rheumatoid arthritis. It's a score, and 15% is what the margin is set up at. But then, so that requires a huge number of patients. But then the similar study on adalimumab, the brand was Exempia, which was from Zydus. So they did the same study evaluating the clinical trial. They enrolled only 120 patients with rheumatoid arthritis, all from India, and they were either randomized to Humira, which is the innovator, or to adalimumab from Zydus. Only 60 patients in each group, and their primary endpoint was also based on ACR 20, which was the case with Remsima or the innovator. And they did it for 30 weeks, the innovator. However, Zydus did it only for 12 weeks. And the margin which was there from the innovator's perspective was 15%. So that is a tight one. Okay. And when it comes to the Zydus one, which they did it for marketing in India, the margin was 28.5. So there's a huge margin. So you even say you will have a huge margin, and so the sample size would be less, and there are high probability that you may be as good as the innovator, and you get a permission. So the Indian regulators did allow this huge margin of 28.5, and they got the marketing permission. But if you go to regulated markets, it would not be possible to get permission with such a small sample size and such a broad margin. There are also studies now we've been designed onto a special type of adaptive designs. I'm not sure if you know what's adaptive design, but I'm sure you guys know Jeff Bezos. So for those who don't know Jeff Bezos, so Jeff is the owner of Amazon. So I think everyone knows Amazon. So with this Jeff Bezos, what is the typical success you could call it as, or what's the typical reason for that type of industry, or what we consider it, like say, for example, mobile phones. So they have been growing pretty fast, right? So probably when I was in my college, we hardly had one company providing mobile phones, and maybe two, and there were probably selective models which were available. So you don't have much of a choice. Now, just say 10 years down the line, or 15 years down the line, we have so many companies preparing mobiles, and those also of an advanced stage. I mean, say when I was in my college, we had to, I would say, all of you who are in faculty would have seen that the keypad was ugly, and so you were not able to easily type. There were no touch phones, and so on and so forth. So the industry has grown. The chip which was there, probably now we have 8 GB RAM, 16 GB RAMs available on phones, which was not the case. We hardly get them on computers at that point of time. So what I mean to say as a bottom line is the industry of microchips has grown in the same period, okay, another 10 years. They have grown drastically. However, when you come to a biosimilar front, or you come to any drugs front, we are hardly able to bring new drugs to the market within this period of time. In fact, we are still struggling with COVID, right? We know COVID since 2 years now, but there is no short drug which could be used for the treatment of COVID. So that's not the case with this industry of mobiles or electronics. So what happens is they have this philosophy of fail fast. I don't know if you know that. So Jeff Bezos also supports that philosophy, is failing fast. So if you stick on to something for a long period of time, and if that fails, all the efforts what you put on goes in drain. So it is essential that whatever you do, if it's going to fail, it should fail pretty fast, so that you get some more time, more cycles to turn up, and then say there could be growth, development, progress. So if as a scientist, if you are in a pharma company, you probably are hardly able to bring a molecule to the market. So by that time you tend to retire. But if you are into an industry of say mobiles or electronics, so there is an upgradation development on a regular basis, which is not the case with us. So we have not accepted this failing fast model, but then gradually we did tend to get into that mode, and we have this model of master protocols. So there are studies now which have been designed with this type of fail fast model in place, and we call it as a master protocol. And that definitely has a drastic reduction in the cost cycle. So there's a huge saving when you come to the pharma perspective. So in case of master protocol, there are two type of designs which are typically possible, three for that matter. There's a basket trial or an umbrella trial. Now, just to give you a fair idea of what a basket is, now what happens is there are multiple diseases, and we try to target these multiple diseases one uh, with one intervention. Like, say, for example, we only bevacizumab for that matter. Okay, so or we already seen adalimumab. So adalimumab is the only drug you have. Now you will also like to see it in plaque psoriasis, you would like to see it in rheumatoid arthritis, you would like to see in ulcerative colitis, and so on and so forth. So with this one drug, you will target 10 indications and see where it's being effective, right? And that's the basket trial. So probably you start the drug adalimumab, you see that it's not being effective in breast cancer, it's not effective in ovarian cancer, it's effective in ulcerative colitis, it's effective in rheumatoid arthritis. Okay, so you screen one drug with various diseases. On the other hand, umbrella trials are certain trials. What happens in that case is you have a single disease. Like, for example, we have COVID at this point of time. So we are targeting all different repurposed drug for COVID, and that's what the target is. So we are trying, say, chloroquine, we are trying whatnot, right? So you know that. So we're trying all different side of bevacizumab also for that matter. So there are various drugs which have been targeted for a single disease, and then you would like to see these type of studies are known as umbrella trials. So you do a study on COVID with say 10 targeted therapies. So if this works, this works, this works, you will try to see. And this is an amalgamation of both. If you put up, it's known as a platform trial. So as the name suggests, for example, you are standing on a platform A, and there is a train which has been coming. Okay, so the train comes from far off places. Say you are in Kolkata, I'm in Ahmedabad. So the train starts from Kolkata and comes to Ahmedabad. So all those who are not showing any efficacy, they would tend to get down at various stations. So you start off with say 10 people on the board of the train, and then you see that okay, this guy is not worth, so he gets down at the next station. And then you find that there's a good guy who could be taken up. So then the next guy boards on at the another station, and he continues the journey unless and until you find him appropriate. Once you find him also not suitable, he takes off the train. There's a new guy joining on the train, and so on and so forth. So there are various analysis which will be done, and those guys who are suitable were the one who would be ending right from, say, you start from Kolkata, you come to Ahmedabad. So there'll be, in this journey will be, there is so many people coming in, going out, and there'll be only few who will be able to reach the final destination. So these type of trials are designed, they're known as platform trials. And there is a huge demand for these type of master protocols in now, and they tend to be very efficacious in getting the drug to the market really quick. And these are a few of the biosimilars who've been tested through the master protocol design, and trastuzumab is one of them, and erlotinib, crizotinib are a few other examples for that matter. So when we come to this master protocol design, we definitely have an advantage with respect to smaller sample size. The hit rates are higher, so you tend to get a hit very fast. And the problem lies more with respect to that is usually a single arm study. We typically don't get to compare it with anyone at that point of time. So multiple testing and study definitely faces several issues. The another important advantage is with respect to the prognosis and the other aspects. The disadvantage is it's usually longer duration. Total screening activity takes a lot of time, but they have been effective, and we have more than 15.5 million survivors with this protocols in place, and there's a huge amount of saving, in fact, in case of time as well as money. So these type of things have been the new thing and in progress. Just sharing, I think we have hit the time almost. Sharing a trivia with you. I'm not sure if you know that penicillin was discovered way back in 1941. At that point of time, it cost about $20 per 1 lakh units of penicillin. And the typical dose of IV penicillin at that point of time was from 4 million to 24 million units is what was supposed to be given. So if we calculate the cost, it would be costing nearly $5,600 for the total treatment for, say, the disease at that point of time in 1941, when penicillin was just discovered. And in that point of time, the cars in the US usually cost around $925, or the rate of gold was only $33 an ounce. So that was the cost of treatment of penicillin at that point of time in 1941. And if we translate that into the current cost, extrapolate that in the 2020 cost, that would be nearly $7,50,000 USD is what would be the cost. And you know the current cost of penicillin, right? We hardly use penicillin at this point of time. We have better drugs available, and the cost of penicillin at this point of time is peanuts. So we assume the same situation would be the case with trastuzumab or adalimumab or any new biosimilar which has been developed. They are expensive at this point of time, but probably a few years down the line, when we'll be re-evaluating these, they would be costing pretty less. So that's it from my end. Thank you. Thank you for being a good audience.
Yeah, thank you, Dr. Jani, for your excellent overview of this clinical, that is involvement of this clinical context of biosimilars, and sharing your expertise and accepting our invitation. I request the participants, if you have any questions, please post it on the questions box.
I think we can close as well. That should be fine.
Yeah. Thank you, Dr. Ashutosh.
Thank you. Thank you for being a wonderful audience. Thank you.
I think we will. I'm closing this session. For the second session, we'll be joining at 11:45.
Thank you.
Thank you all.